Hello everyone, today we are going to present on sickle cell disease, biological pathway and CRISPR-Cas9 gene therapy. Our team consists of Magdalene Tian, Balkis, Changwini, Gretayo and Valerie Gui.
To begin with, sickle cell anemia is an inherited blood disorder that affects the structure and function of red blood cell. This disease is caused by a mutation in the HBB genes, which produce a normal form of hemoglobin known as hemoglobin S. Under low oxygen condition, hemoglobin S molecule tends to aggregate, causing red blood cell to become rigid and sickle-shaped. As this rigid sickle-shaped cell obstruct blood vessel, they cause several serious complications. First, patient develop chronic anemia resulting in persistent fatigue and reduced physical capacity. Second, restricted oxygen delivery to tissue can cause severe pain crisis and joint damage. Third, repeated sickling damage to the spleen, weakening the immune system and increasing the susceptibility to infection. Most critically, this vascular blockage can impair blood flow to the brain, placing patient at a substantially higher risk of life-threatening stroke.
In normal hemoglobin, there are two alpha and two beta subunits that hold the heme molecule. Today, we look into the hemoglobin beta gene, or HBB gene, that encodes the beta subunit. It locates on chromosome 11, and mutation at here will cause sickle cell anemia. An adenine to thymine single nucleotide mutation changes the 6 codon from glutamine to valine. This forms a sickle hemoglobin. Individual with one normal and one mutant gene is identified as sickle cell carrier with no symptoms. In contrast, patients with homozygous mutant genes will suffer from sickle cell anemia. This is because the hydrophobic valine causes abnormal sickle-shaped red blood cell and hemoglobin polymerization, manifesting anemia condition.
After abnormal hemoglobin S is formed, the disease pathway begins when oxygen levels become low. Under these conditions, hemoglobin S becomes insoluble and polymerizes into long, rigid fibers within red blood cells. The formation of these fibers distort the normal structure of the red blood cells, causing them to lose their flexibility and adopt a characteristic of sickle or crescent shape. As a result, these sickle cells become rigid and have difficulty passing through narrow blood vessels. Then, the rigid sickle cells obstruct small blood vessels, leading to vessel occlusion. This blockage reduces blood flow and limits oxygen delivery to tissues and organs. Consequently, oxygen deprivation may cause ischemic injury, tissue damage, and severe pain episodes known as vessel-occlusive crisis. Additionally, sickle red blood cells are fragile and undergo accelerated destruction, resulting in chronic hemolytic anemia.
Imagine a molecular pair of scissors capable of rewriting genetic typos, that is CRISPR-Cas9. In our project, we use this groundbreaking technology to tackle sickle cell disease right at its cellular source, permanently correcting the mutation that causes red blood cells to rupture and collapse. The process begins by harvesting hematopoietic stem cells directly from the patient's blood. In the lab, we introduce a CRISPR machinery. The system precisely targets and knocks out a specific genetic break, the BCL11A gene, which acts as a molecular switch in our cellular blueprint. By disabling this switch, we trigger a brilliant biological workaround, the reactivation of fetal hemoglobin. This healthy oxygen-rich hemoglobin format, which we all naturally produce before birth, take over, effectively bypassing the defective adult sickle hemoglobin entirely. Finally, after the patient undergoes a brief conditioning treatment, these newly upgraded gene-edited stem cells are reinfused back into the body. They successfully graft into the bone marrow, continuously producing healthy, round red blood cells, offering a lifetime cure.
CRISPR-Cas9 therapy provides several benefits for sickle cell disease patients. First, it targets the root cause of the disease by editing the BCL11A gene. Second, it reactivates fetal hemoglobin production, which prevents red blood cells from sickling. Third, it reduces complications such as pain crisis, anemia, and organ damage. Finally, because the patient's own stem cells are permanently edited, the treatment has the potential to provide long-term or even lifelong benefits. In conclusion, sickle cell disease is caused by the mutation in the HBB gene that leads to abnormal sickle-shaped red blood cells. These distorted cells can block blood vessels, reduce oxygen delivery, and lead to severe health complications. CRISPR-Cas9 gene editing offers a promising treatment by reactivating fetal hemoglobin production, reducing red blood cell sickling and disease complications. This highlights the potential of biotechnology to treat genetic disease at their source. That's all from us. Thank you.